and also the mRNA vaccine, which probably, if it hadn't been for the COVID-19 pandemic, we probably are still not sure whether we are going to use it or not. Now, we know that vaccine directly blocks the transmission of pathogens, causing infection, and also decreasing the transmission, the number of infections, and reduces the chance of a pathogen mutating to a drug-resistant form. Although I've heard one of our speakers mention that even though it is the case, but sometimes in terms of vaccination for animals, then it's not always supported. But the world must prepare for the next pandemic caused either by natural agents circulating in nature or by genetic modified organisms created by bad scientists. Hopefully it never happened with bioterrorism. Now we have got so many vaccine platforms, as you have probably noticed during the various presentations, and we know that we have, for example, we have a microorganism here in the form of a virus. Of course, we also have bacteria and other microorganisms, but this is just the example. We see that we have various different platforms here. We can use the whole protein, for example, in this case, the spike protein of the human coronavirus, SARS coronavirus 2. You can also use just a part of the whole protein, so not the whole protein. For example, just the part that binds with the human receptor. In this case, in SARS coronavirus, it's called the RBD or receptor binding domain. And you have the other platforms, such as, for example, attenuated whole virus, like that you replicate, but also you weaken the virus so that it will not be harmful. Yet some of the attenuated viruses, we know they can revert back to become wild-type viruses, becoming pathogenic. The case that we have observed and we also find in Indonesia is the poliovirus, a vaccine that referred back to become a vaccine-derived poliovirus. Now, we also have the whole virus inactivated or killed vaccine. And also we have recombinant vaccine, recombinant protein that can be expressed in E. coli, in mammalian cells, insect cells, yeast cells, and also we can have what we call as viral-like particles. Viral-like particles do not have genetic materials inside it, but it has what we call as repetitive structure. So with this repetitive structure, it can stimulate a stronger, usually it will be towards antibody, neutralizing antibody especially. And we can also use viral vector vaccine. So we can use viruses, either inactivated or replication competent viruses that we have inserted with foreign gene. In this case, the gene, of course, express the antigenic protein. And as the virus infects us, it will deliver the DNA into the nuclear compartment, and then it will be expressed as a foreign antigen that, of course, we expect will stimulate the appropriate immune response, a protective immune response. Also, we have the naked DNA vaccine and also mRNA vaccine. I think with G, that is an inactivated virus vector vaccines that I don't know whether I can here. But you can see that the virus itself may not be exactly, for example, in the case of COVID-19, the virus may not be a COVID-19 virus platform. We can use any virus, but on the surface of the virus, you can have the COVID-19 spike antigen. So it is still stimulating immune response that is protective against COVID-19. Now, the antigen that is potent, we know, should be a protein, and that stimulates immune response towards invading infectious agents. But all of the vaccine platforms should be able to function as an antigenic protein that effectively stimulate protective immune response against invading microorganisms. Now, some vaccine platforms are already available as antigenic protein preparation, for example, inactivated pathogens such as virus, bacteria, etc. We also have live attenuated or weakened pathogens and recombinant protein and viral-like particles. Other vaccine platforms have to enter our cell in order to produce the antigenic protein. And mRNA, of course, is one of them. And also we have the live attenuated weakened pathogen. They can be directly functioning as the antigen themselves, but as they replicate, they will produce more antigen. That if it is a virus, for example, it will be produced in the similar fashion of the virus itself. Like whatever we put in here will be produced in the cell. So if it is a viral protein, then it will be produced just like the virus antigen is being produced as it replicates in our body, the actual pathogen. Now we have the replication incompetent and competent factor vaccine. One of them is not replicating, the other one can replicate, but both again has to be expressed in our cells. And we have the the last one is the DNA vaccine, which has a different mechanism in order for it to be able to be translated into antigenic protein. And of course we have advantages and disadvantages of various vaccine platforms. We have the viral factor based that exhibit highly specific gene delivery into the host cell with strong immune responses. The good thing about viral factor based vaccine, you don't have to handle the infectious virus itself, like what happens when you have to produce a whole virus or even the inactivated whole virus vaccine. With live attenuated vaccine, you will develop long-lasting immunity, high potency, and existing infrastructure used for several licensed human vaccine, and it is said that it is a low-cost manufacturing. You have the inactivated virus as well, and of course, you don't want the large amount of virus to be handled, and antigen integrity needs to be confirmed. So you have the advantages and disadvantages. Of course, I cannot go through all of them. Now we have the RNA viruses. Also, you don't need to handle infectious viral particle. Nowadays, it is low cost, and you even have some of them are even more cheaper to produce and also easy to manufacture. Translation of our mRNA occurs in the cytosol of the host cell, thus reducing the risk of integration into the host genome, which is, of course, contradictory to what happened with DNA vaccine. Now, with DNA vaccine, again, you don't have to handle infectious viral particle. It is even more easy to manufacture, and in comparison to mRNA vaccine, and the synthetic DNA is temperature stable and cold chain free. So this is one of the advantages that we really like about DNA vaccine. However, we have the disadvantages here. The titer remains low, even though it elicits both cytotoxic and humoral immunity. So this is the plus of DNA vaccine, that it can induce cytotoxic immune responses, being able to be produced, the protein being translated in our own cell cytoplasm. And we know that the protein will be chopped by proteases, and then it will be expressed on MHC class 1 molecules. Thereby it will stimulate CD8 T cells. And also one thing that we worry is that it has the potential integration to human genome causing abnormalities. But then I will discuss this later in my other slides. With subunit protein, it can be used in immunocompromised patients and does not involve any live component of the viral particle. Here it is mentioned as having low immunogenicity, but nowadays with the advances in computational biology and bioinformatics, we can actually start to overcome many problems regarding recombinant protein. Now, this is the ideal vaccine, at least according to me and some of the publications mentioned. It can be easily designed to adapt with rapidly occurring mutations that change antigenic structure, short to time and easy to manufacture, safe. It is in mobility, either storage and transportation, and we know that this is mostly due to the stability of the DNA vaccine, and it has long-lasting immunity. Now, this is very important in terms of the long-lastingness, because we now know more that mRNA vaccine that has saved us from the COVID pandemic, actually it is better in stimulating antibody responses, and it cannot be long-lasting. So you have to keep on being vaccinated in a more frequent interval, while in DNA vaccine, it is more long-lasting. So what about DNA vaccine? The advantage of DNA vaccine, we know that similar to other molecular biology-based vaccine, you can design it using synthetic and PCR methods, and you can design easily, make modifications, etc. Advances in bioinformatic technology has given us the ease to be able to design the antigen that we hope can work better. And you can identify regions that has immunogenicity towards B cells, towards CD4 T cells, CD8 T cells, and also regions that has the potential to cause allergic reaction, and also regions that has toxicity, also regions, for example, that can cause you developing Creutzfeldt-Jakob disease, the prime disease, something that we know occurs in the spike region of SARS coronavirus 2. And actually we know that there's an increase in prion diseases after COVID-19 pandemic. And we are beginning to observe that in Indonesia ourselves as well. But with the advances of technology, of course, we can identify this kind of region and try to not include them in our antigen design. The time to manufacture is rapid, and also with formulation, also you have the advantage of having safety and reproducible, no significant adverse events in any clinical trial. So this is one of the advantages which I think are really desirable to individuals, because of course nobody wants to have adverse event after they are being vaccinated. Yet it is stable and more temperature stable than conventional vaccines, and it has long shelf life. Again, mobility and immunogenicity that is long lasting. It can induce antigen specific T and B cell responses similar to those elicited by live attenuated platforms. The DNA vaccine, we have learned that it can happen since 1992, and up to now, we have been trying to develop various DNA vaccine. And we have HIV, malaria. WHO certainly is trying to develop malaria vaccine, and Indonesia is included in the development of mRNA malaria vaccine. But hopefully we can start with DNA vaccine as well. Here it is just showing you that in order to produce the antigenic protein with DNA vaccine, it has to enter the nuclear compartment because transcription occurs inside the nucleus. And from there, it is transcribed into mRNA. The mRNA is translated in the ribosome into protein, and this protein will function as an immunogen. So DNA vaccine has to enter the nuclear compartment in order to function as a vaccine by producing antigenic protein. This slide here is showing you the difference between the mechanism of DNA and RNA vaccine. With mRNA vaccine, you can just deliver the RNA into the cytoplasmic compartment, and there it will go to the ribosome and then be translated into the antigenic protein. While with DNA vaccine, it can be taken by the cell into the endosome, but with that, it may not escape the endosome. So there should be a strategy that we apply so that the plasmid can escape the endosome. So there should be an endosomal escape, otherwise it will be degraded by lysosome inside the endosome. Also, if your DNA cannot be taken into the cell and gain entry into the nuclear compartment, then there's a chance it will be degraded by kinases as well in the extracellular compartment. Now, after endosomal escape, the DNA should be able to get transported into the nuclear compartment, passing through a nuclear membrane. Now, this is what happens if the cell is in non-dividing stage, because in non-dividing stage, the cell nuclear membrane is still intact. So the problem is most of our cells, especially dendritic cells, of course, the cell that we desire to deliver our DNA into, our cells, most of them are in non-dividing stage. So that makes DNA vaccine becoming less effective because you have problem in transport into the nuclear compartment. Okay, so this is just showing all the vaccine platforms for COVID-19, and with DNA vaccine, it was not as successful as the other platforms. Why does DNA vaccine fail? Again, because it has to be effectively delivered into the nuclear compartment first, and the problem is DNA does not readily cross the cell membrane. It needs active transportation. It cannot just leak into the nuclear compartment through an intact nuclear membrane. So even if it is outside of the cell, it has to go through the endosome. So endosomal uptake has to take place, and after that it has to be transported to the nucleus after it escaped the endosome. Now, here we see that there are a long way from extracellular compartment into the nuclear compartment. DNA has to face nuclease, degradation, lipid bilayer, and once it entered the cell via endosome, it may be degraded in the lysosome, so you need to apply some sort of protection here. And then it has to go into the nuclear membrane through, if you notice, there are pores here. And it has to go, it can go through active transportation. It needs what we call as NLS, or nuclear localization sequence. Nowadays, researchers have identified nucleic acid sequences that can act as nuclear localization sequence. But in our work, we try to use peptide sequence that will act as nuclear localization sequence. So it can be in the form of nucleic acid sequence or amino acid sequence. And also there are many strategies that can be applied to enhance immunogenicities. You can select high GC contents and optimize the codon, etc. So many things that you can apply, optimize the gene and avoid certain things. You can get the slides later on because I really have a short time here. For 3 minutes. So, and formulation and delivery. So I think I need to go really fast here. So you see that you can inject DNA and then from the muscles here, it will produce the protein. But also the DNA can get into dendritic cells. And if it gets into dendritic cells will move through the afferent lymphatic vessels, enter the lymph node, and there it can make contact with CD4 and CD8 T cells. The CD4 T cells can be activated because the dendritic cells uptake the antigen in exogenous form, while it can stimulate CD8 T cells because it enters the DNA enters the dendritic cells and express it on MHC class 1. So just showing here that actually DNA vaccine has been shown to actually, if it is given in combination with protein, it can stimulate better T helper 1 and T helper 2 responses. Now also they have tried to use it in macaque and also in duck hepatitis virus, and clearance of the viruses that is used for challenge is actually really good. So it can be used for cancer as well. I need to be really quickly. You can read this yourself. Production, of course, there are regulations. Efficacy and safety, what we worry about integration. Actually, nowadays, WHO has realized that the problem is not as big as we thought. We can discuss that later on if you're interested. And there has been guidelines from WHO, but WHO tried to revise the guidelines again in 2019 because this one is older. Okay, so it is quite safe. If it is delivered into muscle, subcutaneous tissue, or various dermal layers. Right, so I'll just go to my conclusion. Well, this is the work that we have done at the University of Indonesia. We used a peptide that can bind to the DNA and also include sequence, amino acid sequence that we call the cell-penetrating peptide that can penetrate through the cytoplasmic membrane, and also we include the nuclear localization sequence. And if we compare here, using lipofectamine, we didn't get any signal at all. But using the peptide, this is PBMC, plastic adherent human PBMC. Most likely, they are macrophages. So we only get signals using our peptide. This one here just showing you that it is important to deliver the DNA into dendritic cells. And there are lots of strategies nowadays that has been developed so that we can deliver DNA vaccine into dendritic cells. Right. And of course, we can use it for immunization of human and animal. Of course, there may be differences in patient with the nuclear compartment, as well as targeting DNA vaccine to dendritic cells may increase the effectiveness, safety, and quality of DNA vaccine. Ease in design and manufacture. stability and ability to produce long-term immunity are traits of DNA vaccine that has the potential for economical production, storage, dissemination, as well as administration. And this last one is just a recommendation that investment for collaboration between OIC scientists for improvement of DNA vaccine for use in human and animal is beneficial for acceleration to a safe and highly effective, as well as halal platform of DNA vaccine. So with that, I would like to close my presentation. Thank you, Mr. Chairman, for being patient with my presentation.
Thank you very much, Dr. Bala. We don't have a time much, but if there is only one question which is burning very quick, most welcome. Just one question, a quick one.
Can you hear me?
Yeah, I can hear you.
So this DNA vaccines are about 30 years old. So there was much hope about them. But it was said that up to primate level, they are effective. But when it comes to humans, the results are not promising. So what are your comments for this statement?
Thank you. So I think in general, it is better in smaller animals than in larger animals. I think that's the statement that everybody knows. And of course, like primate and non-human primate and human, we are bigger. I suppose the problem is with delivery into into the nuclear compartment of non-dividing cells, because most of our cells are not dividing as quickly as smaller animals. And in laboratory experiments, we do observe that. The other problem that I haven't been able to have time to explain is with the choice of promoter. Now we know that using viral promoters in humans, you may get a transcriptional silencing at the end due to methylation. So we need to still work on those issues as well. In terms of delivery into nuclear compartment of non-dividing cells, I think we are getting better there. There are electroporation systems. In our study, we try to use peptide that I have shown been able to deliver the DNA into non-dividing cells. And actually, I didn't explain, but our data shows that if you immunize mice with the naked DNA, you don't get the immune response as strong as when we are using our delivery peptide. So if that you have the proof of concept that it is indeed important to deliver into the nuclear compartment of non-dividing cells. I think there are many other things that we still have to work on, and that is why collaboration is very important.
Thank you very much. Thank you. So now we have online Professor Jason McLennan from University of Texas, USA. This is talk online, and the title of this talk is Structure-Based Vaccine Design.
Sorry, just a few seconds.
Okay, Dr. Jason McLennan is renowned for his groundbreaking research on viral and bacterial protein. Dr. McLennan's work has played a pivotal role in the development of vaccines and potential treatment for deadly pathogens that have profoundly impacted global health. As one of the inventors of the revolutionary method to engineer key protein in coronaviruses for vaccine use, his technology has been instrumental in leading vaccines against COVID-19, including Pfizer, BioNTech, Moderna, Johnson & Johnson, and Novavax. Additionally, Dr. McLennan's contributions extend to designing crucial protein for vaccines against respiratory syncytial virus, further demonstrating his commitment to advancing public health, honored with the prestigious scientific award and recognized in prominent media outlets worldwide. Dr. McLennan's research prose is widely acclaimed with his expertise in structural biology and biophysics. We eagerly anticipate Dr. McLennan's enlightened insight that will undoubtedly enrich our discussion and inspire innovation in vaccine development. He will be giving a talk.
आपके पास नेक्स्ट वो दोबारा साउंड उधर से लुक करनी पड़ेगी इसीलिए मैं करना चाह रहा था अब यहाँ से आ गया
Should I start?
Yes, please.
Oh, okay. Okay. All right, thank you very much for the invitation to present the research for the warm introduction. So today I'll be talking about our contributions to structure-based vaccine antigen design. So I do have conflicts of interest as I'm an inventor on licensed patents and patent applications related to prefusion stabilized antigens for RSV, HMPV, coronavirus spikes, as well as antibodies that recognize these spike proteins. Okay. So this is a nice overview of the full scope for structure-based vaccine antigen design, we generally start with people who have survived infections. All right, and they've survived because their bodies have produced protective immune responses, particularly antibodies that can neutralize the virus and protect them. And with recent advances in antibody isolation, B cell sorting, we can now sort hundreds or thousands of B cells and antibodies, screen these with functional assays to determine which of those are protective, can perhaps prevent attachment and entry. And then from those hundreds or thousands of antibodies, we can down select to those that are the most potent and maybe have the broadest breadth, whatever properties that we're looking for. And then once we've identified the best antibodies that humans can make against that virus, we then perform structural studies to determine where each antibody is binding, which glycoprotein or viral capsid, how they're binding. And then with that, we have high-resolution three-dimensional structures. We can start to perform protein engineering and design antigens. And so this antibody binds this red part of the antigen. Maybe we don't need the purple part, so then we can computationally graft the red region onto maybe a self-assembling nanoparticle and use that as the vaccine and try to generate a very targeted immune response that will elicit those best-in-class antibodies. It's an iterative process too. We don't go straight to humans, of course. We go into animals first, and we can isolate antibodies from animals and continually determining structures and performing protein engineering in order to optimize the antigen. A lot of what we work on are viral fusion proteins. There's three classes of viral fusion proteins. Today I'll be talking about some of our work on class 1 viral fusion proteins. So these are all synthesized as single chain inactive precursors, and they need to be proteolytically activated by a host cell protease. So here are three, so the ones I'll talk about, some of the most famous: coronavirus spike proteins, HIV-1 envelope, influenza hemagglutinin, and then F proteins from paramyxoviruses and pneumoviruses. And you see, these are all ancestrally related to a common fusogen, and that's more clear if we align it by the protease cleavage site that's absolutely required for function. So the cleavage site here creates a new N-terminus, a new subunit, and that liberates a stretch of amino acids called the fusion peptide, and this will insert into the target cell membrane. So this region is the fusion machinery. This is what's all ancestrally related to a common fusogen, and it contains 4 elements: the hydrophobic fusion peptide, a transmembrane domain that anchors it in the viral membrane, and then 2 heptad repeats that will interact with each other to form a coiled coil. And ultimately, this will result in the viral membrane and the host cell membrane fusing to create a fusion pore and allowing the genome of the virus to enter the cell. Some of these have fusion suppressive caps that sit on top of the spring-loaded fusion machinery. So for coronavirus spikes, that's the S1 subunit that sits on top of S2 and prevents it from prematurely triggering. HIV-1 envelope, you can see that the fusion suppressive cap is quite a bit smaller, but so it's GP120. It still binds protein receptors like CD4, CCR5. Influenza HA1 is even smaller. That binds to just sialic acids on the surface of glycoproteins on the cell. And then there's the F proteins from paramyxoviruses and pneumoviruses, which almost completely lack the fusion suppressive cap. This is how we think of the function for these. So this would be for something like a respiratory syncytial virus, F, or metapneumovirus, F protein. They first fold into what's called a prefusion state. It's metastable, so it's not the lowest energy form that it can fold into. That's the post-fusion form, but it's kinetically trapped. So it's able to stay in this relatively unstable state until something causes it to trigger. So then a triggering event occurs, you get a substantial refolding. The hydrophobic fusion loops shoot into the host cell membrane. This begins to collapse back down into a pre-hairpin intermediate that continues to collapse all the way into a highly stable post-fusion conformation. And at this point now, the host cell membrane and viral membranes have fused. We have a fusion pore, and the cell is now infected as the viral genome enters the cell. So a lot of our work has been to try to structurally characterize this unstable pre-fusion state, and then engineer substitutions and modifications to the protein so that we can produce this as a stable antigen, which is helpful both for vaccine development as well as using it as bait for antibody isolation. So there's many antibodies that target the pre-fusion state and bind and prevent the conformational change. There's also been development of small molecule inhibitors. Quite a bit has been done in this space for RSVF. And I'll tell you a little bit about the work we've done on RSV and coronavirus spikes and how we've stabilized these for use in vaccines. So a lot of this work started back when I was a postdoctoral fellow at the Vaccine Research Institute at the National Institutes of Kwong's lab, collaborating with Barney Graham. And in 2011, I was able to solve the structure of the post-fusion form of the F protein for respiratory syncytial virus. And later, in 2013, I was able to solve the pre-fusion form of the RSVF protein. And to do that, we used a monoclonal antibody that binds to the apex of the protein and holds it in this conformation. So we solved the structure of the antibody-bound conformation of the F protein. So these are trimeric proteins. One protomer is shown in gray, one is in white, and the other one is in ribbons. You can see that the fusion peptide, which is hydrophobic and will stick into the cell membrane, that's in red here. That's stuck inside the interior of the protein to sort of sequester it away from the solvent. We can look at one individual protomer, right? So we'll compare just one protomer between pre-fusion and post-fusion. You can see about half the protein stays in about the same conformation between pre-fusion and post-fusion. This helix loop helix, a lot of this beta sheet, that's all roughly the same. You can get antibodies that are neutralizing that bind to these regions. So sometimes immunizing with the post-fusion form does elicit antibodies that bind to pre-fusion that will bind to the virus and prevent entry. Excuse me. You can also see that at the N-terminus and the C-terminus of the F1 subunit, you get substantial structural rearrangements. So the C-terminus here in magenta flips all the way around to form the outer helix of this highly stable 6-helix bundle. And then the N-terminus completely refolds. So you have the hydrophobic fusion peptide, you have a helix turn helix in red and orange, this beta hairpin in yellow and green, and then this final alpha helix in blue. And all of this transitions into this long helix in the post-fusion state. So this helix flips around, and then all of these elements, including the beta hairpin, refold into this long alpha helix that stacks on what we call the central helix. So it's a really substantial and elegant conformational change that these proteins undergo. And so once we had these structures, we then had to design substitutions that lock the protein in this conformation and prevent conversion to post-fusion, so we can use it as a vaccine immunogen. We were able to do that through an iterative process. We identified a disulfide bond that we could introduce. It's normally 2 serine residues that are hydrogen bonding via their side chains. We can mutate those to cysteine. And now that forms a disulfide bond where one cysteine is in the region in blue, that undergoes the conformational change, and one cysteine is in a region in gray that stays the same. So that really covalently locks those together. Then we added some cavity filling substitutions to help keep this region folded, particularly this serine 190 to phenylalanine. So a smaller amino acid to a large one fills a pocket and prevents that region from refolding. And so we could produce this, we call it DSCAP1, when my colleague Barney Graham, and they immunized rhesus macaques. So this was a prime and boost, and then serum was drawn later. And so we look at the week 8 neutralizing antibody titers, so higher is better. What we can see is that the post-fusion form of the F protein elicited neutralizing antibodies titers in the hundreds, whereas with pre-fusion F, we are now in the thousands, high thousands, including some in the 10,000. So a really substantial increase in neutralizing antibody titers immunizing with the pre-fusion form of the protein. So this work was all published in 2013. It was a runner-up for the breakthrough of the year. For proof of concept, proof of principle for structure-based vaccine design. In 2015, then, for the next several years, I continued working on pre-fusion F. This was a collaboration with Janssen, which is part of Johnson & Johnson, and Johannes Langedijk. We came up with an alternative way of stabilizing the protein, so some different substitutions. One of the ones I wanted to focus on is this substitution of a serine to a proline in this loop region. And so this region needs to refold into a coil, this coil needs to refold into a helix and stack on alpha 5. And so Hans created this S215P. And if you look, they tested every amino acid at position 215 to proline. And so on the left, we're looking at protein expression. So changing that one serine to proline gave a really substantial boost in protein expression compared to the serine. And if we look at the fraction that retains the pre-fusion conformation, even at letting the protein sit for 18 days, almost all of it retained the pre-fusion conformation. So this targeted proline in the region between the base helix and this first helix that refolds give a big boost in expression, and also, most importantly, helped keep the protein in the pre-fusion conformation. And so that's what we really liked and would use later. We then went on to show in 2016 a basis for why immunizing with the pre-fusion form of the F protein resulted in such high neutralizing antibody titers. And so for this work, we collaborated with a company called Adimab. And together we were able to isolate, produce, and characterize 364 monoclonal antibodies directed against the RSV-F protein from 3 adult donors. And so we could bin these. We created 6 antigenic sites 0, 1, 2, 3, 4, and 5. And we could bin the antibodies into those sites. And what we can show is that the most highly potent antibodies, about 60% of those, targeted site 0 and site 5. And those antigenic sites are only present on the pre-fusion conformation. They are completely refolded and adopt a different structure in post-fusion. So if you immunize with post, you can get antibodies against sites 4, 2, and 3, but you won't elicit any antibodies against 0 and 5, which we're showing are about 60% of the most potent ones. So this provides a nice basis for why you want to immunize with the pre-fusion form, where all the antigenic sites are present. So this is the pre-fusion F protein that we made in 2013, and several of the other ones from Janssen and Pfizer that are based on that work. Those were licensed and started going through clinical trials. In 2022, GSK and Pfizer both announced positive phase 3 data for these pre-fusion F proteins in people aged 60 years and older. And then last year in May, the FDA approved the first RSV vaccines, Arexvy, for individuals 60 and older, and GSK uses exactly our pre-fusion F protein. Barney is shown here receiving his vaccine in fall of last year, holding on to the pre-fusion RSV F protein that is being injected into his arm. So we were pretty excited by that. And so far, for all the data, it's looking very positive. And so then we were wondering, will this work very well for RSVF? Can we apply structure-based vaccine design principles to coronaviruses? This was right as the time I was leaving the Vaccine Research Center to start my own lab at Dartmouth College in 2013. And that was when the Middle East respiratory syndrome coronavirus, MERS, had emerged in Saudi Arabia and the surrounding region. And it was really quite scary because it causes an infection with a high case fatality rate, the high 30 or 40% case fatality rate. And that was the second coronavirus to spill over in a decade. We had SARS in 2002, SARS in 2002, and then we had MERS in 2012, and so we thought we should really learn how to make coronavirus vaccines for a potential next pandemic. So our goal was the same. So this was our plan back in 2013 for coronavirus structure-based vaccine design. Again, collaborating with Barney Graham. We needed to get the high-resolution structure of a pre-fusion coronavirus spike, use that to engineer stabilizing substitutions and lock it in the pre-fusion form, and then test that as an immunogen. So when we started working on this, we were trying to work with the MERS coronavirus spike, but it expressed really poorly. We could make smaller fragments like the N-terminal domain or the receptor binding domain, or even the full S1 subunit. If we tried to express and purify the full spike ectodomain, we essentially got no protein. Fortunately, Barney's lab is also working on the spike protein from HKU1. That's one of the 4 seasonal coronaviruses. And that spike protein behaved much better. And so we could add a trimerization motif or not, and those looked very similar. We could allow the protease activation between the S1 and S2 subunits or not. That didn't really matter. Ultimately, we went with this construct where we mutated the protease site, added a trimerization motif, we got a very nice peak, good expression, and we sent this protein to Andrew Ward's lab at the Scripps Research Institute. And Andrew's lab was able to obtain a 4-angstrom cryo-EM structure, revealing the first pre-fusion confirmation of a human coronavirus spike. So S1 subunit is shown here in blue. One of the S2 subunits is in pink. The others are in gray and in white. See that the N-terminal domains are located on the periphery of the spike protein. The receptor binding domains are mediating the trimerization. There's also this intercalation where the S1 comes down and then back up. The structure revealed 2 new smaller domains or subdomains, SD1 and SD2. And then importantly, it gave us the confirmation of what the spring-loaded pre-fusion form of the S2 subunit looked like, which actually looked quite a bit like the RSVF protein, which makes sense because they're ancestrally related. And so since that first structure, many other structures have been installed by my lab, Andrew's lab, David Visto's lab, and many others, with and without receptor, with antibodies. And so we now have a nice model for how this functions. So we know that the S1 subunit sits on top. The receptor binding domains can hinge open in order to bind to the receptors. That causes instability, and the S1 subunit sheds. And then you have this harpooning and refolding of the hydrophobic fusion peptide into the target cell membrane. You have the pre-hairpin intermediate, and then you get the zippering as the 2 heptad repeats begin to walk towards each other, eventually forming the post-fusion conformation and causing the fusion of the viral and host cell membranes. So these are very impressive little molecular machines. We wanted to stabilize them so we could immunize with the pre-fusion form. All right. And then this shows some of the conservation between the pre-fusion S2 and pre-fusion RSVF. They both have a central helix in blue that does not undergo much conformational change. And then you have the elements of the first heptad repeat in green, yellow, orange, red. All of that refolds into this long alpha helix, very similar to what we saw from the RSVF protein. So we tried several different things. It was a little tricky. We had the structure of HKU1, but we were trying to engineer the MERS spike, and those only have around 35% sequence identity. So some regions are not particularly well conserved. This region is, though, between the central helix and that first helix of the heptad repeat. So you can see that between all the different beta coronaviruses like HKU1, MERS, SARS from 2002, they all contain RLD, 2 non-conserved amino acids, a conserved glutamate, and then a lot of the central helix is conserved. And so we remember that trick from Hans Lagerdyk, where we put a proline into this region. And so my postdoc, Nianchuang, started replacing amino acids on the cap, the central helix, with proline. And what we can see is that just a single proline gave a huge boost in protein expression. And if we put 2 prolines at these 2 positions, we got about a 50-fold increase compared to the wild type spike and the 2 proline forms. So we really like this. We call this the S2P for 2 proline. We could then scale this up. And if we look at wild type versus 2 proline based on gel filtration, again, we can see looking at area under the curve, the 2 proline spike, a huge amount of protein expression compared to this very small peak here in red. The protein was well behaved. The wild type spike existed in both the post-fusion and pre-fusion conformation. And if you let the protein just sit there over time, more and more of the pre-fusion goes to the post-fusion, whereas the 2 proline was just all pre-fusion. It was very well behaved. We set the protein to Barney Graham and Kazmecki Corbett, and they immunized mice at a low dose, a 0.1 microgram dose, and then tested the elicited neutralizing antibody titers. So these are the IC90 titers. Higher is better. They tested either the 2 proline form in red, wild type in blue, or the S1 subunit in orange. And what we can see is that against many of these different spike proteins from MERS coronavirus isolates, immunizing with the 2 proline form elicited substantially higher neutralizing antibody titers than did immunizing with just wild type or S1 subunit. So we had all this published in 2017, which we're very excited about. Well, because this region is so well conserved among beta coronaviruses, we could show that putting those 2 prolines in that same spot for the SARS protein from 2002, we also got a very nice boost in expression. Wild type SARS was a mix of post and pre. 2 proline SARS was all pre-fusion. It also worked for HKU1, which was already quite stable and allowed us to get the structure, but the 2 prolines helped. We showed it worked for many different beta coronaviruses. So this seemed to be a universal approach for stabilizing beta coronavirus spikes in their pre-fusion conformation. And so then in 2018, my lab moved to the University of Texas at Austin so we could have access to cryo-electron microscopes. And so we were well prepared then for when SARS-CoV-2 emerged in late 2019. In early 2020, Chinese researchers had identified a beta coronavirus and isolated the beta coronavirus causing these pneumonia outbreaks. On January 10th, they released the genome sequence of what's now known as SARS-CoV-2. Within 10 days, my postdoc Nianchuang had clones several different plasmids for the spike protein containing the 2 proline mutations. And then 10 days later, a grad student at the time, Daniel Rapp, had purified the spike protein, started cryo-EM data collection, and by mid-February, we had already submitted a manuscript to Science and BioRxiv describing the cryo-EM structure of the proline stabilized spike. So we were able to go from a genome sequence released online on January 10th to a submitted manuscript describing the structure of the stabilized spike protein in about a month. Our spike protein active domain, the prolines correspond to residues 986 and 987, so that's where we put those. Again, we were able to see a nice boost in expression compared to wild type with those two prolines. The protein expressed okay. We were able to get about 1 milligram per liter. This is the structure. We were able to see one of the receptor binding domains in green was in the up conformation. The other two were in the down state. This was published very rapidly by Science. We're trending on Reddit. It's been cited over 9,000 times, which is really exciting. And then what we're most excited about, of course, is that all 4 COVID-19 vaccines authorized for use in the United States use the pre-fusion stabilized spike. So Moderna has the 2P mutations at positions 986 and 987, as does Pfizer and BioNTech. Johnson & Johnson tested several different forms of the spike protein in non-human primates and showed that the 2 proline form elicited much higher neutralizing antibody titers than the other forms, as well as Novavax also used the 2P mutations at positions 986 and 987. Maybe I'll just go very briefly through this so that there's time for questions. We did end up developing a 2nd generation spike protein. We designed over 100 substitutions now that we have the structure, including prolines, disulfide bonds, salt bridges, cavity filling mutations. This work was led by my postdoc, Qinglin Shea, and grad student Jory Goldsmith, along with collaborators at UT Austin. We ended up testing each of these 100 variants and saw several substitutions that boosted expression 6-fold, some boosted 3 to 4-fold. We started combining all those, and when we combined 4 additional prolines, we got a very nice boost in expression of the original 2 proline form. Area under the curve, so combo 47 has 6 prolines, so we call it hexacro. We got about a 9 to 10-fold boost in expression over the 2 proline form. We increased the melting temperature, and by negative stain, it looked very good. We could scale this up. Instead of getting 1 mg per liter with the 2 proline, we are now getting around 11 mgs in one cell line, 32 milligrams in a different cell line, again, much higher than the 2 proline form. We solved the cryo-EM structure to show that it was almost identical to the 2 proline form in white, compared to hexacro in green. And this plasmid has now been distributed to over 215 labs around the world. Barney's group immunized mice, and at every dose, the 10 microgram, 2 microgram, and 0.4 microgram, hexacro elicits higher neutralizing antibody titers than does the 2 proline form. And it's particularly noticeable at the low dose, where the 6 proline is much better than the 2 proline stabilized spike, elicits substantially higher neutralizing antibody titers. It's been incorporated into 2 different, actually several different vaccines. One is based on Newcastle disease virus. That's now being produced and it's been approved in Thailand, as well as in Mexico. It's produced in eggs, so it can be produced very cheaply. And then also the purified hexacro is being administered on a patch, on this small patch that contains 5,000 microprojections. And you can coat the protein on these little spikes, and then you just stick it onto your arm. It self-administers, and it's stable at room temperature for months, which could be very nice for low and middle income countries. So to summarize, the first pre-fusion RSV-F-based vaccines were approved in May in 2023. All approved COVID-19 vaccines in the US contain or encode the stabilizing prolines, and now we're starting to see hexacro-based vaccines that have been approved in Mexico and Thailand. And so this work is highly collaborative. I've highlighted many of the people, particularly Barney Graham, who we've worked on for both RSV and coronaviruses, are funding. With that, thanks very much for your attention, and I'd be happy to answer any questions if there's time.
Thanks a lot. Over
ऑन कर दीजिएगा माइक
My song.
Thank you, Dr. Jason.
Thank you.
नहीं नहीं आवाज जा रही है. It's okay.
तस्वीर भी देख लें.
Sorry.
Thank you, Dr. Jason, for giving a wonderful talk. I will ask any question the audience has, and then I communicate to you.
Okay, that's good.
Yes, please.
Thank you. I'm Faria Hassan from Lahore University of Management Sciences. I'm a PhD student. And thank you, Dr. Jason. It was really nice listening to your work, and it was really, really nice. I really enjoyed it. So, okay.
Thank you.
So my question is, you mentioned that you pre-fusion and post-fusion, especially using pre-fusion confirmation of the state of the spike protein as the vaccine and everything. But now, recently, there have been emerging variants, and there have been more like subtypes of the Omicron lineage. So now they're like saying that there's more differences in the sort of the pre and post fusion. Now there's more difference in that as well. Like it's changing, it's mutating as well. So what's your take on that, and how should we work towards solving that thing, because it's emerging as another issue here. Thank you.
So Dr. Jason, they are saying that what about variants? So we need to produce a new protein?
Yeah, great question. So, all that work was done with the original isolate, the Wuhan or Washington, and then as you mentioned, it's just been a considerable amount of variation in new variants. At the beginning, we were making different forms. The prolines continue to work well for all of the variants, and so we made alpha, beta, gamma, delta. We went up to Omicron. Kind of stopped because it's a lot of work. But the 2 and the 6 prolines seem to work very well. And we've continued to produce those proteins. We've seen in the US and Europe, some of the vaccines are getting reformulated to now include the variants. And so those new boosters do contain still the 2 proline form of the variants. The extent to which we need to continue to reformulate the vaccines to include variants is sort of unknown. We've seen, it seems like humans that have been infected or vaccinated several times now have pretty good immunity. Like we've seen new variants emerge, like the JN1 variant, in a large increase in cases. Infections, but not large increases in hospitalization. So humans seem to have a pretty good immunity now, and we'll have to see at what frequency we need to continue to reformulate those boosters.
Thank you. Any other questions? Yes, please.
Dr. Jason, thank you very much. Very nice, interesting, motivating talk.
Thank you.
I'm a protein scientist working from Christian College, Chartered University in Lahore. You have demonstrated and people have benefited from your research. So I want to ask you a question about foot and mouth disease virus. It's monomer, it's not protective, or its higher oligomeric form, is protective. So can this proline, many prolines be introduced there? What's the principle that would make it?
Yeah, great question. So for foot and mouth disease, that's not one we've worked on, and I believe that forms a capsid. So HPV, human papillomavirus, for the capsid, fully formed is important. Presumably, some well-placed prolines could also work. In addition, just the general approaches, cavity-filling substitutions, disulfide bonds, all of those could work to just help increase the thermal stability of the capsid and keep it together. And we've shown that if you think of the example between the 2 proline and 6 proline form of spike, the 6 proline form, which is more stable, is a better immunogen, and we've sort of seen that as we stabilize proteins, increase their melting temperature, they become more immunogenic. We think that's because maybe more of the protein survives and gets to the germinal center after it's been injected. So for something like foot and mouth disease, I do think there's opportunities to perform structure-based design and identify substitutions that will lead to higher protein expression as well as greater stability of the full capsid.
Thank you very much.
Thank you very much. Actually, some work in Fulbright Institute and Oxford University is going on, and they stabilize it.
I thought I've seen papers from like Dave Stewart's lab has done a little bit there. Yeah, that looks very promising.
Absolutely. Thank you. Any other questions? Looks like no. Thank you, Jason. It's really great to see you.
Thanks again for the invitation. Have a great rest of the conference. Bye.